A comparative analysis of enzyme linked assay. Four types of enzyme linked assay are there: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Among these four types of ELISA, which ELISA used to detect the antigen and which is for antibody detection, then which is more efficient, which is more sensitive? So all these things we will discuss in detail in this video. So hello friends, I am Jumishri Meher, PhD from Banaras Hindu University. So in this video, starting from the definition to its principle and all about the ELISA we will discuss here. So without delay, let's start the video. So first the definition. ELISA, it is a solid phase enzyme immunoassay and colorimetric test used for detection of antigen or antibody, providing both qualitative and quantitative result. First one is the solid phase enzyme immunoassay. Solid phase, why it is called as solid phase? Because here in ELISA we use a micro titer plate. Okay, this is a rectangular plate of 96 wells. It contains 96 wells, and in that wells actually this antigen as well as the antibody will be coated, and then the reaction will occur. So on a solid surface, this enzyme, antigen and antibody are coated. That's why it is called as solid phase enzyme, because this total reaction are depend on enzyme reaction, because the enzyme will react with substrate and it will give some color change, and by that color change only we are detecting the antibody as well as the antigen. That's why it is the solid phase immunoassay, because the immunological bodies are involved. That's why it is known as immunoassay. It is based on antigen antibody reaction, the specific reaction between antigen and antibody. So now you got this point, that why it is called as solid phase enzyme immunoassay, and it is a colorimetric test. As I have already told you, here the color change is detected. The enzyme as well as substrate, they will combine and form some color, and that color are detected in this assay. That's why it is known as colorimetric test for detection of what? Either antigen or antibody. So both antigen can be detected or antibody can be detected, providing both qualitative as well as quantitative result. Qualitative means it will just detect whether some antigen or antibody is present or not, and quantitative means it will measure the concentration of that particular antigen or antibody. So generally this color intensity will directly proportional to the amount of antigen or antibody. So initially this assay was discovered by Peter Perlman and Eva Imgel 1971, later popularized by Waller et al 1970s, you can say 1976. Okay, but in most of the MCQ based books you can find that this Waller name is there. Means generally for the plant assay, means plant based ELISA assay, this name is popular. Means for the plant or for the plant pathogens, this ELISA technique was popularized by Waller et al 1976. Next, about the principle of ELISA. So first, in the microtiter plate well, the antigen or the antibody will be coated. Then the antigen and antibody, they will bind specifically, and one antibody is actually, that will be the enzyme linked. Means any enzyme, alkaline phosphatase or horse-radish phosphatase, so any enzyme it will be linked, and then further this substrate will be added. So if means reaction between antigen and antibody is there, means the enzyme will be there. So when we add the substrate, the enzyme and the substrate will combine and give the color change, and that color development will be detected through spectrophotometrically, that is the colorimetric detection. So if color develop, means this is a positive result, means the particular antigen or antibody is present, and the intensity of color will give the idea about amount of antigen or antibody. So this is about the definition, discovery, as well as the principle behind enzyme linked immunosorbent assay. Next, about the substrate and enzymes. So here the table for the common substrate and enzymes used. This substrate and enzyme are also very specific, means the combination is very specific. The first one is the p-nitrophenyl phosphate, the substrate, and the enzyme used for this is alkaline phosphatase. Here phosphate means definitely phosphatase will be there, and the color it forms is yellow color. So in a ELISA reaction, after coating the antigen and antibody, if the secondary or primary antibody is linked with this enzyme alkaline phosphatase, and we are using the substrate P and PP, then definitely the color that will form after the reaction will be yellow in color, and that will have to measure at 405 nm wavelength. You have to remember all these things, means this commonly used substrate as well as enzyme, as well as this what color this enzyme will form and at what wavelength it is measured. This is most common one. Okay, the first one is the most common one, the mostly used for ELISA assay in different laboratories, commonly used. So remember this first one, and the second one is also most important because of its high sensitivity. If you are considering the sensitivity, you will use this second one, and but the commonly used one is this first one. So second one is the substrate is tetramethylbenzidine, TMB is the substrate, and enzyme used for this is horseradish peroxidase. It will form the blue color at what wavelength? 450 wavelength. These two are enough actually. You can remember this also, but these two are must. You have to remember both the color also as well as the wavelength also. So apart from this is also penicillase. This is also important, but these two are most important. So principle I have already told. The antibody is generally enzyme linked. So if antibody and antigen reaction is there, means enzyme will be there only. So when we add the substrate, it will form the color product at the particular wavelength. So this is about the enzyme and substrate. Next, about the types of ELISA. So 4 types of ELISA are there. First one is direct ELISA, second is indirect ELISA, third is sandwich ELISA, and 4 is competitive ELISA. So in the first, the direct ELISA, this is the most simplest form. Only one type of antibody is used, that is the primary antibody. So an antigen is coated on the plate and detected by enzyme linked primary antibody. Simple. So first here you can see here in the well the antigen is coated, then it will be washed with buffer to remove the excess antigen. Only the coated will be remain there. Then the enzyme linked primary antibody will be poured over the plate to interact with the coated antigen, then allow it for some time to bind, and then again wash it to remove the excess unbound primary antibodies. then the substrate will be added. So if color is developed, means that particular antigen is present. Okay, and if color is not developed, means no antigen is there. Means actually antigen and antigen are the foreign molecule, the viruses, mostly virus protein, as well as the fungal protein, or any proteins are there for any foreign pathogens, whereas the antibodies are immunoglobulin proteins that specifically detect the antigen and activate the immune system. Okay, so we have to design a particular antibody for a particular type of antigen. The reaction is very specific, they are very specific, they act very specifically. Okay, so if any color change is there, means the our concerned antigen is there, or our concerned virus is there. So we will read that intensity of the color change. By that we can detect the amount or concentration of that particular antigen. So features are, it is a very simple and rapid method, because simply just put the antigen, then antibody, primary linked enzyme linked primary antibody will be given, then substrate will be added, finally color developed, and we will read the absorbance. The less step or less time, because only single antibody is used. In other processes more than one antibody or antigen are used. Then but it is lower sensitive than indirect ELISA. How? See here, only one time we are confirming, means the antigen is there, and one time only we are confirming by adding the primary antibody only. We are not cross checking it another time, no further step is there. Simply we are checking the color development by adding the substrate. No secondary antibody, no cross check is there. That's why the sensitivity is lower as compared to the indirect ELISA. Then each primary antibody must be conjugated with enzyme. This is also important, because if some primary antibody are not conjugated, means it will not properly give the result. Okay, so this is about the direct ELISA. Next, second type is indirect ELISA. This direct ELISA is for antigen detection, whereas this indirect ELISA is for antibody detection. So indirect ELISA, here antigen is coated on the plate, detected by primary antibody followed by enzyme linked secondary antibody. Here one single step is added in indirect ELISA, that the antigen is indirectly detected by the secondary antibody, and the secondary antibody is enzyme linked. Means first the antibody will be coated on the plate or the vessels, then the primary antibody will be added, but the primary antibody is here not the enzyme linked. And the further cross check the presence of antigen, we will add secondary antibody that will bind to the primary antibody. Okay, and that is enzyme linked also. Then the substrate will be added, color develop, similar things are there. Okay, only one extra step is use of a enzyme linked secondary antibody. So here feature is, first feature is first feature is the higher sensitivity, definitely, because this method two times we are cross checking, first through the primary antibody, second through the secondary antibody, so it is of high sensitivity. Then more economical, because here one enzyme labeled secondary antibody can detect. See, the secondary antibody will be same for all this antigen. Okay, only we have to change the primary antibody. That's why it is more economical as compared to the direct method, because in the direct method we have to design the primary antibody and it should be enzyme linked. But here the secondary antibody is universal and it is enzyme linked, so we don't have to take tension about enzyme linked antibody. It is universally present, means it can be universally used for all this antigen. Only we have to take tension for the designing of the primary antibody. So it is more economical as compared to the direct ELISA. Then third is longer time, more step than direct ELISA. Yeah, definitely, because one extra step, the step number 3, another antibody we have to use, so it will take longer time and one more step than the direct ELISA, but it is more widely used for antibody detection. So this is about the secondary, direct and indirect ELISA. Then the third type is sandwich ELISA. So sandwich ELISA, here the antigen is captured between two antibodies. A captured antibody coated on the plate and enzyme linked detection of antibodies there. So in the direct and indirect, what we did, we are coating the antigen over the wells, but here antibody will be coated. So primary antibody initially coated over the microtiter plate, and the primary antibody is not enzyme linked. Okay, after coating the primary antibody, then we will put the antigen, and then after reaction with antigen and primary antibody, we will add the secondary enzyme linked antibody. Okay, so here also two antibodies are there, two antibodies are used, but the sequence are different. In case of indirect ELISA, the sequence is antigen, antibody and antibody, whereas in case of sandwich ELISA, it is antibody, antigen and antibody. Okay, just this difference is there. Then this substrate will be added and color change will be detected spectrophotometrically. Then features are, first one is it is very high sensitivity and specificity. Okay, then best for detecting low level of antigen. As you can see here, the sequence is very peculiar. It is, the antigen is trapped between the two antibody, that's why it is very high efficient in detecting low level of antigen also. Then require two antibodies, the primary and secondary antibody for detection. Then ideal for complex samples like extract, sap, serum. So this is most widely used. Then third one, the fourth one, that is competitive ELISA. So it is simple antigen compete with an enzyme labeled antigen for binding sites on a limited amount of antibody. So in the first one, indirect ELISA, one antibody, one antigen used. In the indirect and sandwich ELISA, we use one antigen, but two types of antibodies are used, primary and secondary. But here in case of competitive ELISA, two types of antigen are used. One is our the concerned antigen and another is the reference antigen, that is enzyme linked. So one is normal antigen used and another is enzyme labeled antigen is used. So these two actually compete to bind to the antibody. So first the antibody will be coated on the plate, then both this sample antigen as well as our enzyme labeled antigen, both will be added to the sample, to the plate where already the antibody is coated. So what happened? They will compete for the binding site. So if the more fluorescence or more color if you get, means our enzyme linked antigen is more present, our sample antigen is less. So the relation is opposite, means darker the color, less antigen are there, because we don't need our enzyme linked reference antigen is not our concern antigen. Our concern antigen is the unlinked one. That is the major pathogenic agent or pathogen virus antigen, that is only. So because that can develop any color, so the relation is here indirect. More color is there, means more color, if more color is developed, means less sample antigen is there, and if it is developing light color, means more sample antigen are there. This is about the types of ELISA. Then the advantages and disadvantages of ELISA. First, the advantages. It is a rapid method because total 96 wells are there, so many samples can be run simultaneously. Next, it is of high sensitivity. So actually its sensitivity is 10 to the power minus 6 nanogram, I think. Check it. Yeah, 10 to the 6 nanogram level. 1 nanogram level, I think. Please check this. If you know, please mention it in the comment section that what is the sensitivity of ELISA. It's 1 nanogram, I think. Yeah. I remember this. It is 1 nanogram. For the sensitivity of ELISA is 1 nanogram and sensitivity of PCR is 1 picogram. Okay. Remember this fact. This is very important. Then the reaction is very specific. It is very high specific because of specific antibody and antigen interaction. Then it requires very small sample volume because small wells are there. So only 10 to 100 microliter sample is sufficient. Then is high throughput. It is a high throughput technique. Then it is widely applicable for detection and diagnosis. Next, the disadvantages. So it requires specific antibodies. Then cross reactivity may occur because non-specific binding or false color development may be there. Then false positive, false negative result may result because if you are not doing proper blocking means to block the unspecific binding, we are removing the unnecessary unbound antibody or antigen. Then require proper washing. Then enzyme and substrate stability issue is also there, and initial cost is higher. So these are the disadvantages. Finally, the application part. So application, first is it is used for detection of plant viruses, seed health testing also we can do. Then certification of the virus free planting material, like indexing program, it is used in certification schemes also it is used. Then it is used for large scale disease surveillance and research application for quantification of virus load, as well as for host plant interaction studies it is used. So this is the comparative analysis for the 4 types of ELISA. Let's recall all this. So direct ELISA, here directly the antibody is quoted, then enzyme linked and primary antibody will be given, then substrate, then color development. In the indirect ELISA, first antibody will be quoted, then over that primary antibody will be given, then secondary antibody, that is enzyme linked, will be added, then substrate added, then color developed. Then in the sandwich ELISA, the antigen will be sandwiched between primary and secondary antibody, and then substrate added, color developed. Then and in the last competitive ELISA, the antibody is coated over the plate, and 2 types of antigens are used: sample antigen and enzyme linked and enzyme labeled antigen. So they will compete for the antibody, then substrate added, the color change will be studied, and here the color change is indirectly proportional to the presence of antigen. Then about the sensitivity, Sensitivity is highest for the sandwich ELISA and lowest for the direct ELISA. The number of antibodies used. This is important. Here in the direct and competitive ELISA, only one antibodies are used, whereas indirect and sandwich ELISA, 2 antibodies are used. Steps, similar things only. Then best use, direct ELISA mostly used when high antigen level is there, because it is of very low sensitivity. Okay, that's why high antigen concentration must be there. Then for indirect ELISA, best use is for antibody detection. It is a general purpose. Sandwich ELISA, when the antigen concentration is low, we generally prefer the sandwich ELISA. This is the best one. Then competitive ELISA, for quantification of antigen, it is inversely proportional. Okay, so this is all about ELISA. So now you are very much clear about ELISA. So if you have still any doubt, you can mention that in the comment section. So thank you for watching this video.